My name is Vanessa and I would like to present the introduction of bacterial pathogenesis. Bacterial pathogenesis is the process by which bacteria cause disease in a host through various mechanisms such as transmission, colonization, invasion, toxin production, and the development of symptoms. Pathogenic bacteria use specialized structures such as fimbriae, pili, and adhesins to attach to host tissues and evade the immune system, and also taking advantage of weak points while avoiding defenses like phagocytosis and complement. Phagocytosis is an immune cells that eats bacteria, while complement is a protein that destroys bacteria. Understanding this process is essential for developing effective treatment and vaccines.
My name is Debbie Deborah Denisius, matrix number BS23110016. Today, me and my friends Heli Halim will be present about objective. For objective, first, to identify major bacterial pathogens. The main bacterial pathogens in aquaculture include Vibrio, Aeromonas, Streptococcus, Edwardsia, and Flavobacterium. These bacteria are normally present in the environment, but become harmful when condition allows them to multiply or when the fish immunity is weak. Second, to examine environmental and management factors. Environmental and management factors play a major role in triggering outbreaks. Poor water quality, high temperature, and excess waste create ideal for bacteria to multiply. High stocking density, frequent handling, poor biosecurity, excessive feeding, and introduction of infected seed further increase the risk to fish. Therefore, it is encouraged to regularly monitor the environment and management of fish to reduce mortality and excessive consumption costs.
The third objective is to analyze clinical signs and pathology. This means identifying what symptoms the fish or shrimp are showing when they get infected by bacteria. It includes observing behavior changes like reduced appetite, slow movement, for the external signs, lesion discolorations, gill issues, and for the internal damage, organ swelling, and hemorrhage. The goal is to understand how the disease affects the animal's body so we can link specific signs to specific pathogens. The last one is to develop strategies for outbreak investigation and response. This focuses on creating a clear plan to detect, manage, and control disease outbreaks in a farm. It includes steps like collecting samples, identifying the bacterial cause, checking water quality, reviewing farm practices, and tracing where the infection started. The aim is to respond quickly with the right actions such as treatment, biosecurity measures, and preventive strategies to reduce losses and stop the spread.
Hello, my name is Muhammad Hazrul Hilmi and my part is methodology. First, study sites and sampling, for example, sample size, sample tissue, and water sample. Second is clinical examination and water quality check, such as pH, DO, and others. Third is bacterial isolation and identification, which is PCR, 16S, RNA sequencing. Fourth is virulence assessment, such as hemolysin, protease, lipase activity, biofilm formation. Fifth is antimicrobial susceptibility testing, AST. Sixth, histopathology, and lastly is data analysis. That's all from me.
My name is Amanda, my metric number is BS23110163, and today I will be talking about the results and the key findings of our poster. So the first part is the major bacterial pathogens, which include Aeromonas hydrophila, Aeromonas salmonicida, Edwardsia ictaluri, Edwardsia tarda, and Flavobacterium columnar. As for the environmental and management factors, the first factor is stressful conditions. So in stressful conditions such as high population density, changes in temperature, and hypoxia, it can actually accelerate the spread of pathogenic bacteria. The second factor is biosecurity. Inadequate biosecurity measures can actually increase the risk of disease outbreaks. And the third factor is genetics. When genetics is not taken into account when selecting fish for aquaculture, it can lead to reduced productivity, increased vulnerability to disease, and environmental stress.
The third objective is clinical sign and pathology. Example of clinical sign is fish is inactive and show poor appetite. Second, red lesions on the skin. Example of pathology: first, swollen liver; second, pale or damaged gills. The fourth objective is outbreak investigation and response. For example, check the water quality. Second, separate sick fish into a quarantine tank. Third, take samples for lab testing to confirm the disease.
Hello everyone, my name is Edrina Lawrence, matrix number BS231-60392, and I'll be presenting on the discussion. Based on RPE Yanong et al. 2025, major bacterial pathogens such as Aeromonas edwardsii and Flavobacterium are important causes of ulcers and also gill damage in farmed fish such as tilapia and African catfish. Intensive aquaculture conditions favor rapid spread of these pathogens, leading to disease outbreaks and major economic loss. Some of the conditions that may attract disease is high stocking density and also poor water quality.
Hello, my name is Ningmatulay Mintia Abdulkarim, number matrix BS23110066. For the discussion, long-term strategies should emphasize good husbandry practice, regular monitoring of environmental parameters, and strict biosecurity protocol. IND Robertson, 2020. This measure not only reduce pathogen introduction but also limit transmission within and between culture systems. Next, research advance improve vaccine and probiotic tools play important role in disease control. Kandarkarasima Tukaram, 2025. Ultimately, strengthening management practice and deepening our knowledge of fish pathogen interaction remain the foundation for reducing outbreaks and promoting resilient aquaculture system. That's all for me. Thank you very much.
So in conclusion, bacterial pathogens, particularly Aeromonas edwardsii and Flavobacterium, are major contribution to disease outbreak in aquaculture, often leading to symptomatic ulcer and gill damage in culture fish. Environmental and management factors such as poor water quality, stressful condition, inadequate biosecurity, and weak genetic selection further exacerbate this outbreak. Understanding bacterial pathogenesis, identifying key pathogens, and recognizing early critical signs are essential for developing effective prevention and control strategies. So although limitations such as sampling, constraint, and variability of field conditions were encountered, this finding emphasized the need for improved farm management, stronger biosecurity practices, and continued research to support sustainable aquaculture health. A stronger focus on pathogen monitoring and proactive disease management can significantly enhance fish survival, farm productivity, and long-term aquaculture resilience. That's all for me. Thank you.